Good morning, everyone. I'm Simran Da Costa, and on behalf of Altum Technologies, I'd like to welcome you all to this virtual event on plant-derived secondary metabolites for the treatment of diseases. So, before we begin, a little bit on secondary metabolites derived from plants. So, we see that despite the recent contemporary researches in the field of allopathy medicines, especially when we talk about synthesized pharmaceutical agents, nature is still the main supplier of bioactive molecules, especially from plants to cure diseases like cancer, infections, and other neurodegenerative diseases. And most of these bioactive molecules are secondary metabolites like alkaloids, flavonoids, steroids that are derived from fruits, roots, barks, and leaves of plants. So, although the usage of plants in treatment of diseases began with the traditional treatment regimes, we see that plant derived compounds tend to cover a large portion of the pharmaceutical agents in the development of modern medicine. For instance, Withaferin-A, which is a steroidal lactone compound, is derived from Witania somnifera and is used to treat SARS-CoV-2 infections, cardiovascular diseases, hepatocellular carcinoma, and 13 other types of cancers. Similarly, we see that resveratrol from berries, curcumin from turmeric, quercetin from fruits, vegetables, leaves, seeds, grains are used to treat various kinds of cancers. Now, in bioinformatics, virtual screening of plant compounds for a particular target is a very efficient method for screening lead compounds from the existing phytomolecule databases like NCBI PubChem in computer aided drug design. In addition to that, drug protein interactions provide insights on the network of interactions of pharmacologically active natural plant compounds in pathway databases like StitchDB. And these secondary metabolite plant based therapeutic compounds are efficiently used for the treatment of diseases by deploying smart targeted delivery methods like mitochondrial targeting and nanoencapsulation. So let's understand more about plant-derived secondary metabolites for the treatment of diseases from our speaker of the day. It gives me immense pleasure to introduce Professor P.K. Praveen Kumar. So, Professor P.K. Praveen Kumar is presently working as a professor in the Department of Biotechnology at Sri Venkateshwara College of Engineering. He's got more than 20 years of experience in teaching and research in various engineering colleges in Chennai. He's completed his BTech in chemical engineering in VIT Vellore, MTech in biopharmaceutical technology at AC Tech, Anna University, Chennai. His PhD in biotechnology in SVCE under Anna University, Chennai. He's published more than 30 papers in reputed international and national journals, mostly related to anti cancer drug discovery, antiviral drug, and vaccine design. In addition to that, he has published three national patents in three book chapters. He has worked on plant based adjuvants of dendritic cell vaccine for the past 13 years. He has mentored more than 50 UG projects and 9 PG projects and is presently guiding 3 PhD students in the areas of anti cancer drug design for hepatocellular carcinoma, drug design for type 2 diabetes. He has also done consultancy projects for Tata Institute of Social Sciences, Mumbai, Sri Krishna Arts and Science College, Coimbatore, Welltech Rangarajan, Dr. Shagantala R&D Institute of Science and Technology, Chennai. He is a PhD supervisor in Anna University, Chennai, and doctoral committee member at Welltech Rangarajan, Dr. Shagantala R&D Institute of Science and Technology, Sri Ramachandran Institute of Higher Education and Research, and Anna University, and PhD board examiner in University of Madras, Chennai. He is presently working as the principal investigator in a DST SERB Government of India funded project worth rupees 18.3 lakhs on checking the efficacy of Vitanolide A Warfarin A plant-based compounds derived from Witania somnifera on mitochondrial targeting of heat shock protein 90 on hepatocellular carcinoma and protein bioinformatics lab, Indian Institute of Technology, Madras, Chennai, since 2021. He is also working as a team leader in UT 2.0 innovation funded project of rupees 5 lakhs on Vitanolide A, which is a potent inhibitor of mitochondrial HSP90 of hepatocellular carcinoma. He has received a Young Scientist International Travel Grant Award from CSIR ICMR and National Travel Grant Award from SERB in the areas of pharmaconoxy, more specifically in plant based drug design for diseases. He has also received grants to conduct national workshops, seminars, faculty of development programs in the areas of artificial intelligence, machine learning approaches in drug and vaccine design, in cancer from CSIR, DBT, ICMR, AICTE, Government of India. He has also associated with Altum Technologies Bangalore to organize a 15-day certificate course in CADD in 2021 CSIR sponsored workshop on genomics, data science, and precision medicine in 2021 and AICTE sponsored FDP on artificial intelligence and machine learning approaches of drug design in cancer. So, welcome once again, sir. And I'd like to hand over the platform to you.
Thank you very much, Simran, for a detailed description about me. So I think the PPT is visible to everyone. Is it so, Simran? Okay. Is it visible? Can I go ahead on presentation?
Yes, sir. Yes, sir. You can go ahead.
Good. Thank you. Yeah, so good morning to everyone, to all the participants here. So myself, I am Professor P.K. Praveen Kumar from Department of Biotechnology, Sri Venkateswara College of Engineering. I first like to thank Altem Technologies as well as Department of Biotechnology, Sri Venkateswara College of Engineering to provide me an opportunity to present on the title Plant-derived secondary metabolites on the treatment of diseases in the virtual event organized on the importance of natural products in therapeutics. Yeah, so these are some of the organized events linked with Altem Technologies. So from CSER, I have received a fund of nearly 25,000. Okay, but also we got some kind of a sponsorship to conduct a kind of a workshop on machine genomics, data science, and precision medicine design, which is related to cancer. So getting a secondary metabolites treatment on cancer. And the second is the certification course, also linked with Altem Technologies. So 15-day certificate course, majorly relying on the computer-aided drug design. And we have got a funding from the DBT Government of India to conduct popular lectures in biotechnology. And recently, 4 months before, we have received yeah, from AST of amount 3.5 lakhs to conduct an FTP, that is AST Atal FTP on unraveling molecular mechanisms and artificial intelligence approaches of drug design in cancer. So now let us go to the title of the presentation. So I'm here to present about secondary metabolites. So first go to the importance of plants in life. Why we have to consider importance of plants? See, not only herbivores, also carnivores has to dependent on plants. If not directly, we have to dependent on indirectly, isn't it? So all each and every organism has to rely on the plants for food. Not only for food, even for the pharma, for the drug purposes, for drug or vaccine, even we can rely on the plant sources. So how can we rely on the plant sources? We'll be seeing this in the subsequent slides. Before going to the importance of plants and its compounds, let us go to this impact of health rings, the advertisement which was popularly spread all over India last month. What is that? The Ministry of Commerce and Industry has instructed all the e-commerce platforms to remove beverages such as whatever mentioned, okay, whatever shown here, from the classification of health drink. Okay, now we can't call this as a health drinks, not only in the e-commerce platforms, but also in the stores. We can't able to keep a title like a health drinks for this. What is the reason behind? The reason behind why they have banned to keep this title is it's having more sugar, and even some of the chemicals are not up to the desired level. So obviously, just because we are having more sugar content, okay, now it's and also most of the people are facing from the autoimmune disease like diabetes. Now they try to remove this title. Then what we can consider that as health drinks, just shown in the next slide. So an extract from plant compounds, I mean plants like plant seeds, okay, cereals, Okay, this is what we are calling it as a health mix, isn't it? There are so many companies they are coming up with a navadanium and some other combination of grains. Okay, now we can term this as a health mix. According to that, just an extract searched in Google today. I'm showing all these things as a health mixer. So what you can understand, obviously, from the plant sources, we get rich nutrients, not only for the food, also it can take even that in the form of a drug if you take it in desired quantities. Now let me take you to this next slide about the metabolites. So what are the classification of metabolites? The major classification is primary metabolite and the second one is the secondary metabolite. What are primary metabolites? These are molecules that are essential for growth and development of an organism. So most of the large biological macromolecules like carbohydrates, proteins, lipids, hormones consider the examples of primary metabolites. Then if you see the secondary metabolites, okay, the toxins, secondary products, nature products derived from that, including amino acids, antibiotics, derived from bacteria, fungi, animals, and plants. All these things which are not directly involved in the normal growth or development. Okay, this we call this as secondary metabolites. Now let me take you to the major difference between the primary and secondary metabolites. So if you see, I already mentioned the primary metabolites are required for the growth and maintenance of cellular function. But whereas in secondary metabolites, these are just involved in maintaining the ecological functions. And primary metabolites occurs at the growth phase, and secondary metabolites occurs at the stationary phase. Primary metabolites produced in large amounts and easy to extract, but that is not the same case for the secondary metabolites, only produced in small amounts, meager amounts, and difficult to extract, of course. For an example, whatever compound I'm using in my research, okay, so just 10 milligram of compound cost, the price comes around 16,000 to 40 or 45,000, just 10 milligrams, more than the expensive for diamond. Okay, so getting a secondary metabolite is not a easiest task. Even if you get with 95% purity and more than that, obviously you have to spend a lot. Okay, and if you see the next difference in the primary metabolite is that it is same in every species. Okay, whatever metabolites we extract is almost same. But whereas in secondary metabolites, each and every plant has its own kind of uniqueness. Okay, of secondary metabolites. Okay, I'll be taking you to the slides where you can differentiate the plant sources and the kind of secondary metabolites secreted from that. Now, next point is it perform physiological functions of the body. Here, there is these are derivatives of primary metabolites, isn't it? So obviously, you can get physiological functions accordingly. And the best examples for primary metabolites or carbohydrates, vitamins, the lactic acid, etc. But for secondary metabolites, you can see the phenolic steroids, antibiotics, pigments, etc. Now let me take to the next slide about the secondary metabolites. How the secondary metabolites are derived? These are just derived from again from the primary metabolites. Okay, so the secondary metabolites are majorly derived from primary metabolites. For this, I just showed you a kind of a flow chart. Okay, here in this flow chart, you can able to see a derivative compound called pyruvate is originated from the glucose. How you can form the pyruvate is formed from the glucose only through the glycolysis cycle, isn't it? Similarly, if you see erythrose 4-phosphate, this is a derivative we got from the glucose from the pentose phosphate pathway. And if you see the secondary metabolites which are shown in this pink color, okay, isoprenoids. If you say similarly, isoprenoids are produced from the malonic acid, or even if you see the major raw material is the glucose, of course. But if you take from the glucose, it is produced from glycolysis cycle. It's also passing the TCS cycle. So combination of cycles, pathways are involved to produce this kind of isoprenoids. Similarly, if you take flavonoids, which is the major drug for the cancer and a few more diseases, okay, the flavonoids or the complex flavonoids are formed from the hydroxybenzoic acid or aromatic amino acid, but basically these are of course formed from the glucose. So if you see the flavonoids are formed from the pentose phosphate pathway, cycumate pathway, etc. Okay. So likewise, you can able to find the secondary metabolites or majorly derived only from primary metabolites. Let me take you to this slide about the classification of the secondary metabolites from plants. There are majorly four classification of secondary metabolites. Number one is phenolics and polyphenolics. You find a list of compounds like phenolic acid, flavonoids, stilbenes, coumarins, tannins, lignans, curcuminoids, xanthones, naphthoquinones, acetonophynones, all comes under phenolics and polyphenolic compounds. The second classification is terpenes and terpenoids. So these are the examples: monoterpenes, sesquiterpene, diterpenes, triterpenes, tetraterpenes, polyterpenes. The third major classification is the sulfur-containing compounds, alanines, glucosinates, phyto. These are a few examples. We have plenty of examples. Okay, so just showing whatever the author Ajay et al. Has exposed. And for the nitrogen-containing compound, free alkaloids, seed alkaloids, protoalkaloids, polyamide derived alkaloids, etc. Now let me take you to the next slide. About the databases of the natural plant compounds, there are plenty of databases. I just showed you the major databases what we use. Okay, number one is the natural product catalog from the zinc. This is the largest database everyone know. Not only we have the repository for the synthetic molecules, but also we have this is also a repository for the natural products. So for this, you have to go to this link. Similarly, IMFAT. Okay, I think even initially the madam has explained in the first lecture. That is, IMFAT is the national database for the Indian medicinal plants, and majorly it tells about the phytochemistry and therapeutics. Okay, so anyone doing research in Ayurveda, Siddha, Unani, all these areas of medical research, taking the botanicals, taking the plant compounds as a drugs. Okay, obviously you can go in search of this IMFAT database. Okay, and I think Bureau of Indian Standards, Ayush, etc. Also linked with the TBT and DSD coming forward to give a large number of grants, isn't it? So if you want to work on the kind of a grant projects, obviously you have to look through these kind of a database. Initially, you can work on informatics, and then you can try to work on the extraction, purification of the compounds, and even to try to use these compounds for in vitro studies and in vivo studies for the treatment of any disease. Okay, so let us see what are the major databases. Number three is the Lotus. Okay, of course, you can find more natural products here and Molpot, Supernatural 3.0. For plant secondary compound database, we have an exclusive website called pscdb.appsbio. Dr. Duke's phytochemical and ethnobotanical databases. Okay, there's also a large kind of an international database from and PDTDB, that is phytochemical and target database. For this, the URL is mentioned: Natural Products Repository, DNetDB, and NPMRD. Now let me take you to the next slide about the steps and techniques required during the characterization of plant products. So generally, whatever environmental sample you have collected, okay, even from my side, I just working more on ashwagandha. Ashwagandha in India, but according to the genus name, we call that as a Withania somnifera. Okay, that is an Indian ginseng plant. For cultivating this Indian ginseng, I bought the seeds from the natural remedies from Delhi. Okay, I purchased that from the Delhi. I got the seeds and I just cultivated this in SVC soil, and I'm using these plant roots and the other parts for the extraction of the compounds and trying to work for various biological activities. Okay, so if you try to cut any kind of an samples, okay, you just have to that is can be a root sources mostly for the extraction of drug compounds, but sometimes we can also focus on the other parts like stem, even leaves, okay, barks, and especially the fruits and seeds are also taken for the extraction of drugs. So in that case, whatever sample you collect, you go for fractionation of the crude extract. Then you have to screen based on the bioactivity assay. If you're focusing on a particular protein and a particular biological activity that is for the respective disease, you have to first screen it accordingly. Then you have to take an active extract, then go for secondary screening and identification, go for large scale production and purification. Even the large scale production purification includes utilizing the plant fat technology, okay, using the plant fat technology, that is plant tissue culture, okay, and you can also try to increase the content of this particular secondary metabolite, and you can extract accordingly according to the optimized method of extraction and purification procedures. If you see the de-replication methods, okay, for the first screening by activity assay, these are most of restriction fragment polymerism methods, HPLC methods, fingerprinting methods are generally used for the first screening by activity assay. And for the de-replication databases, you can also use antibase, merandit, antimerin, actibase, ifinter, comet, etc. So now let me take you to the next slide about each and every compound. Okay, now we discuss each and every secondary metabolite and its significance of that compounds. Okay, first we can go to the popular compound paclitaxel. This we also call it as a Taxol. Okay, generally this is the active compound found in Taxol, and it is used as a chemotherapeutic drug, majorly used to treat cancers like from ovarian. breast, lung, capillary sarcoma, cervical cancer, and pancreatic cancer. These are the types of cancers majorly treated by this compound Taxol. This is the first significant anti-cancer compound, first isolated in 1973 from the barks of a coniferous tree, the Pacific view. Let us go to the next compound, resveratrol. This is the compound, this is a type of stilbenoid, a natural phenol compound found in skin of grapes, blueberries, raspberries, mulberries, and peanuts. And this is majorly used for treatment of breast cancer, osteosarcoma, cervical cancer, colorectal cancer. I just showed you a kind of a case where like we can work with these kind of corresponding cell lines, and already they have reported this is significant in apoptosis for the breast cancer and the osteosarcoma, and there is compound show significant results in autophagy for cervical cancer and apoptosis for the colorectal cancer. So the findings also shown obviously. Now let us go to the third compound, curcumin. I think everyone know about the curcumin. This is a principal curcuminoid of turmeric, also called as Curcuma longa, a member of the ginger family, Gingerbaceae. So this is majorly used for treatment of various types of cancer, but I showed the major cancers like melanoma, skin cancer. Here you can find the autophagy. So we are treating this melanoma through autophagy, and breast cancer, again autophagy, cervical cancer. We can try with the HeLa cell lines. This already reported results. Also tried with few more cell lines. I showed just an example. Can be through apoptosis. We are this works on apoptosis and colon cancer, obviously, again through apoptosis. If you see the curcumin biosynthesis, you know the curcuminoid, or what we call it as a curcumin, okay, is synthesized only from the phenylalanine. You got it? So this compound, not only this compound, even most of the compounds, whatever I've shown, resveratrol and other compounds, if you see the biosynthesis pathway, okay, it might have been synthesized from an amino acid, most probably an amino acid, or a kind of an organic acids. Okay, so curcumin is of course synthesized from the phenylalanine. Now go to the next compound, quercetin. This is again a popular cancer drug. Okay, this is a plant flavanol derived from this also comes in the flavonoid group of polyphenols and majorly present in fruits, vegetables, leaves, seeds, grains, and red onions. And majorly it is used to treat pancreatic cancer, cervical cancer, and colorectal cancer. Now go to the next compound, ginsenoside. This is, of course, we have, I think, only three ginsenoside derivatives. Okay, if you want to find the major derivatives of each and every compound, can be curcumin, resveratrol. Simply go to the NCP Pumpkin and you just check it out. Okay, we have also derivatives of compound. I think ginsenoside is a major compound. Okay, out of the other two derivatives, and majorly used to treat breast cancer and the lung cancer. So the mechanism and findings are shown here. Now go to the next compound, Withaferin. So this I am working on Withaferin compound research for more than 12 to 30 years, more than 12 years. So if you see the description, this is steroidal lactone compound. Okay, Withaferin, the structure is shown here. Is derived from, it's a steroidal lactone derived from ashwagandha, that is Withania somnifera. The roots are shown here and the leaves are shown here. If you see this, this is majorly used to treat more than 13 types of cancer, but I reported only two. It has already reported to cure 13 types, and the 14th type I just illustrate. I'm just working on. That is, I'm working actually on liver cancer, especially on hepatocellular carcinoma. It is also showing a kind of a good results with the hepatocellular carcinoma. So I think we'll be getting a few publications with it. Okay, so the cell lines and the mechanism and findings are shown here for this two types of cancer. So now I'm just showing you a kind of innovations happened funded from the Ministry of that is Institution MHRD, IAC, Institution Innovation Council. Okay, the Institution Innovation Council has conducted a competition called Drug Discovery Hackathon. Okay, in 2020. So actually, we have totally four phases, but the Department of Biotechnology, Stephen Gretschmer College of Engineering students have came up to the finalist in phase one, and they secured nearly 3.6 lakhs. Okay, so they, the problem statement they provided to work is on identifying covalent spike protein inhibitors to control the infection of SARS-CoV-2 from small molecule databases. You know what I did? For this problem statement, I have taken the sample, the input from the NCBI PubChem. I've downloaded the secondary metabolite information from the NCPA PubChem, and I started working on with a kind of a methodology. Okay, so this is the event we won actually, nearly 3.6 lakhs, and subsequent in the next year, okay, we just my PhD scholar just competed and another kind of event called the India Smart Protein Innovation Challenge. This is organized by Good Food Institute, but the earlier was organized by CACR, AACTE, MHRD, Innovation Cell, etc. Okay, so Good Food Institute has selected only 20 projects. Out of these 20 projects, this is the project with the top score. She secured first prize of 1.07 lakh. Of course, we worked on plant-based selection of functional algae and their fractionation to create suitable proteins and fats safe food product application. Now, let me take you to the methodology of what we worked in drug discovery hackathon. That is how we have used to utilize the secondary metabolites. Okay, so this is the methodology I propose. So generally, we have downloaded 1,152 compounds. You know, during the pandemic situation, okay, everyone got confused, including clinicians, researchers. They don't know what drugs has to be prescribed for the COVID-19 infection. Okay, especially everyone are aware that even India is declared pandemic in March 2019, isn't it? So when the people who initially infected with the COVID-19, what has happened? If they approach the clinicians, they give a combination of 8 drugs, whatever drugs they provide, okay, or a repurposing drugs of anti-cancer activity. So they have provided drugs for dengue, that is, they provided anti-dengue drug, anti-malaria drug. They also provided a kind of that is dengue, malaria, chikungunya. They have taken all this, okay, but they also given HIV drugs. See how the HIV is integrated with this kind of a COVID-19 infection, but they don't have an idea. They want to cure. Okay, that's the one day objective they have, and so they have treated with even the HIV drugs. But it resulted any kind of a good results. We won't get a good results, isn't it, by using the repurposing drugs for a new kind of viral infection. So what the government of India has initiated, especially MHRD, IAC has initiated a kind of a competition, not only for the students, but also for the scholars and the faculty members and even the industrialists. So even the industrialists came forward to provide softwares to use freely for 3 to 4 months, I believe. So we utilized a few softwares, okay, which they hosted in online. We have utilized these kind of softwares, but myself, I didn't utilize it for this drug discovery hackathon. For drug discovery hackathon, I utilized only the freewares. Okay, of course, we utilized the kind of databases and tools available in online. And for docking, I utilized Autodoc, and for simulation, I've gone for the Promax. But even the Discovery Studio from BioVia also came forward to offer free services during that time to come up with a solution. So I thank even Altem Technologies and the overall the BioVia Discovery Studio persons who has given offered this support. Now let me discuss about the methodology of what I proposed. So during that time when I gone to the NCPA PubChem, okay, only 1,152 compounds are there, okay, which are present as a data set for treatment of COVID-19. So I just downloaded all the 1,152 compounds and checked for the ADMET properties and drug likeness properties. Okay, I just screened some 400 base compounds from that, and then I screened the 100 base compounds through the virtual screening through Autodoc PYRX. Then whatever I screened, 100 base compounds, I just filtered based on the doc scores. Okay, based on the energy scores, I filtered only the top 10, and I just taken further for docking using Autodoc. Okay, I just docked all the 10 compounds, and I checked not only the binding energy scores, but also I checked the binding sites, whether is it binding the appropriate binding sites of the spike glycoprotein. The spike glycoprotein is a very large protein because we'll be getting nearly 1,333 amino acids is the length. Okay, and initially we didn't try it with the kind of strains like alpha, beta, gamma, delta, but we just tried with the initial spike glycoprotein, which is unmutated at that time. Okay, tried with it, and but the problem statement is to try with the open and closed states of spike glycoprotein. So we have tried with these two types of protein, and we finally came up with the best three compounds generated from the docking, and whatever three compounds we got, we have screened. Okay, we also taken for the simulation, and we also finalized and validated which is the best one. So when we got after the docking and simulation results, we have received Withaferin-A, abivatinib, and luteolin, or the best secondary metabolites, okay, used to treatment of COVID-19 infection, that is to target SARS-CoV-2 spike glycoprotein of both open and closed states. But if you compare overall the docking and simulation, the Withaferin has showed maximum as docked with a spike glycoprotein with maximum binding sites, nearly docked 4 out of 13 binding site amino acids. So we have concluded Withaferin is the best compound. Not only I've just concluded this from the computational tools, even Professor Sundar, okay, I believe Professor Sundar from IIT Delhi, he also has proposed the same, okay, during his research, and also this kind of proposed innovation he also published in newspaper like The Hindu, okay. So the same result, of course, I got fortunately during my computational study results. So I can mention you that even the computational study results, we can rely on the computational study results before developing the drugs and going for the in vitro and in vivo activities. Now let me take you to the next case study. So this research published in a journal in 2022. We worked on actually this is a UG funded project, okay, from Rajalakshmi Engineering College, a faculty named Rekha. She has secured a grant, and we of course worked on various computational parts. Not only we worked on computational, I think she also worked on in vitro and other studies. So this is a research article. Actually, we have utilized molecular docking. We have tried with Autodock and MD simulation using Chromax, and pharmacophore modeling we tried with PharmaGist, and rescoring protein ligand complex through DSX scoring. Okay, again we got Withaferin-A. Is a potent secondary metabolite used to treat the cardiovascular disease. Okay, so for this Withaferin-A is already shown antiviral activity, and now you are getting an information that is also used for treatment of cardiovascular. And I also going to show one more study that is also used for treatment of cancer, liver cancer, especially hepatocellular carcinoma. I also received multiple grants on it. Now, let me take you what exactly we worked in the article I showed previously. So, the docking results were not only just interacted with Autodock, but we also go for drug score extended scores. Okay, we have received, and we just finalized our Withaferin is binding with the top two binding receptors. We have taken the top two binding receptors, and then we also have gone for the MD simulation. And whatever interaction nature DSX scores we got, showing we have just shown these scores in bar diagram. So a list of hydrogen bond interactions also shown in that journal paper. Now let me go to the next secondary metabolite, Withanol A. So till now whatever I discussed is about the Withaferin. So Withaferin, you have plenty of derivatives, Withaferin A and hydroxywithaferin A. There are plenty of derivatives of Withaferin A. Similarly, even withanolide, you have nearly 35 types of withanolides. Okay, not only you can get this as a derivative from ashwagandha, we also have few more plants, Indian plants, where you can get these compounds. You know, the Withania somnifera, what we call it as an Indian ginseng, it belongs to the tomato family, Solanaceae family. Like a tomato, even you find these berries looks like a tomato shape. Okay, also we can get withanol from the berries, but majorly we extract much content only from the roots. Okay, so also I'm working on these withanol compounds for more than 12 years. And I just reported that this compound shows better activity on breast cancer and lung cancer, and the findings on the mechanism is shown here. And now let me go to the next slide. Okay, so this is just an extract from that showing that I'm working, not only I'm working as a professor in Department of Biotech SVC, but also presently actively working as a postdoctoral research fellow. Okay, as a principal investigator, I have received 18.3 lakhs. So I'm working under a mentor, Professor M. Michael Gromiha, who is India's #1 bioinformatician in India. So methodology proposed in Sartare project. Okay, so actually we have proposed to not just taking the Withaferin-A for the treatment of cancer, but we try to I try to utilize utilize the mitochondrial targeting. I just integrated the mitochondrial targeting regions with the linker. So I've synthesized nearly five important derivatives, Withaferin-A derivatives, and whatever derivatives linked with the mitochondrial targeting regions. This I've used it to target a target receptor called HSP90, heat shock protein, which is most widely expressed during the cancer. Okay, during the cancer, you can find HSP90 expression is more when compared to the normal patient. So this made me to precisely target. Okay, so how I'm precisely targeting, not only I'm using this as a protein biomarker, but also the mitochondria is generally present more in the cancer cells when compared to the normal cells. So this is a major motive. So I just want to target only the infected tissues. If you find a liver, okay, if we have two lobes, isn't it? If one lobe is infected and if the other lobe is not infected, we have to ensure during the chemotherapeutic treatment, whatever drug we have, technology we have used, okay, the drug has to target only the infected lobe, not the healthy lobe. So this is my objective, to reduce the pain and to target efficiently with less dosages. I target just to develop this technology. Of course, even this technology can overcome the existing drug sorafenib. The sorafenib is major used drug for treatment of cancer, especially the liver cancer. If you take, it can treat the third stage of cancer. If you go to the last stage, okay, it can be the cirrhosis or hepatocellular carcinoma, which is the final stage. We can't able to treat. Okay, so during these conditions, the patient can better go for transplantation. So, but up to the third stage, we can able to cure, but with effective chemotherapeutic and radiotherapeutic techniques. Okay, so from my side, okay, so whatever technology developed to produce this kind of Withaferin-A derivative or Withanol derivative, this can be used for the treating the third stage of the cancer. So I just not only tried with the computational studies with the system biology approach by getting the gene expression and profiling, which is most closely interacting with the HSP90. Okay, and also I tried with the molecular docking approaches. I've gone for the simulation, and also I just completed in vitro studies, that is especially mitochondria isolation and mitochondria assessment. All these studies are completed in vitro, and now we are presently working on in vivo studies. My PhD scholar is working extensively on in vivo studies of these Withaferin-A derivatives in HSP90 of hepatocellular carcinoma. Now let me take you to the next case study, the paper published on hepatocellular carcinoma. This is another compound, Withaferin B. The Withaferin B is a retinoid derived from Withaferin diffusa. Withaferin diffusa. Actually, this plant is generally used for rejuvenation. Like if a person's or having a urinary infection, or if they have the kidney stone failure. Okay, so this compound is giving a better remedy. Okay, but now I tried with whether is it playing significant in hepatocellular carcinoma or not. Yes, it's working well when I tried with the computational studies, when I compared with the controlled standard drugs used for treatment of hepatocellular carcinoma. I just cross-checked whether this compound is showing a significant score when compared to sorafenib and other drugs. Yes, it's showing, obviously. So what are the techniques I used here? I used molecular docking like Autodoc, NMD simulation using Chromax to find to prove the result. So these are some of the hydrogen-bond interactions. Not only I tried with one or two, I tried with nearly five receptors. Okay, for based on the different mechanism of cancer action. So here you can able to find the binding site, binding energies are shown, and the binding site interaction sites are also shown here, which is reported in this journal paper. Now let me take you to the next innovation. So this innovation, actually, whatever Praveen be, I checked for computational studies. We also cultivated this Withaferin diffuser, and we have whatever routes we have taken, we just isolated and purified up to 92 to 93 percentage. So for this, we have used the chromatography techniques. Okay, and after using the chromatography up to the HPLC and after purifying up to 92%, I have characterized the functional groups using FDR. So whatever we purified, the compound is taken further for checking the stages of dendritic cell development. Okay, myself, I've worked on dendritic cell development by using the plant compounds as plant-based adjuvants. I just check what is the effect of these plant-based adjuvants on the stages of dendritic cell. Fortunately for the Withaferin B is increasing the maturation and activation of the dendritic cell. Especially it is increasing maturation. So this I just determined through the FACS analysis through dendritic cell markers like CD80, 86, and 80. Okay, so this kind of innovation I completed during my PhD research. Now let me take you to the case study 3. So here I just used different plant-based compounds, of course, the secondary metabolites for the treatment of cancer. Here majorly I used system biology approach. Okay, majorly I used system biology approach, and the databases we have used Cake, Funcook, GEO, Panther, etc. So whatever case studies I am reporting, these researches happened in this decade. Okay, from 2020 to 2024. Okay, so here if you see these are the databases used, and the data set used is the cancer. The cancer tissue samples are around 267, and the healthy volunteer samples are around 18 in a data set. So we have taken a data set, and we have gone for extensive uh, gene expression analysis through system biology approach. And not only we just enrich the genes and identify the top 10 genes during the approach. Okay, we used a workflow and we just screened the top 10 genes. And after enriching the top 10 genes, we just interacted with the top 3 plant compounds. And we checked which is the best plant compound in docking. Okay, this is how we concluded. So this is how we found out the potential herbal target for the ovarian cancer. So whatever 3 compounds I mentioned, these are generally used plants for the treatment of ovarian cancer. Okay. So and if you see the major tools I have utilized is the MATLAB, Cytoscape, and SwissDoc. Now let me go to the next slide. So about the results of the extract, what published in Informatics and Medicine Unlocked in Elsevier journal. So majorly I just gone with the 1,000 important genes, actually 500 upregulated and 500 downregulated. Then I screened up to top 100 upregulated and downregulated based on the scores. Okay, and finally we have gone according to the pathways, the secreted pathways, and then the disease targeting pathways, and then we filtered based on the scores. Okay, we have filtered only top 10 genes, and whatever top 10 genes we have taken, we have just talked with the 3 plant compounds like apigenin, quercetin, and resveratrol. And of course, we got the quercetin has shown negative maximum energy for 6 genes out of 10 genes. Out of 10 genes, we got 6 positive results. The negative maximum binding energy result for quercetin. So we have finalized that quercetin, flavanol, is the best compound for treatment of ovarian cancer when compared to apigenin and resveratrol. So this is a case study for this. Actually, we just worked even a few compounds extracted from Mimosa pudica. Okay, so the databases we use is PubChem and PDBsum. Technology uses molecular docking, and these are the tools we have used. We relied majorly on Discovery Studio, of course. Of course, even Sri Venkateshwara College of Engineering is having a product, Discovery Studio. Secondary metabolites from Mimosa pudica. So these are the 5 plant compounds we have replaced in research, and mimosin is the best compound we got from docking and simulation. And molecular docking results of secondary metabolites of Mimosa pudica with a particular receptor. So we have shown some kind of docking energies, and we have generated from Discovery Studio. So we got plenty of scores like Lick score 1, Lick score 2, PLP1, chain scores. Okay, so dock scores, internal energy scores. And we also got the interacting binding amino acids. Now let me take you to the next secondary metabolite called anthocyanin. So now presently I'm working on this anthocyanin. Okay, the anthocyanin comes under a group of flavonoids, majorly found in black rice, black soybean. So most of the black berries, black cabbages. Okay, so in all these things we find plenty of anthocyanin. My PhD scholar is presently working on this research. Okay, she is working on both type 2 diabetes and as well as a kind of a liver cancer. Okay, and of course, we just initiated on computational part at present. So already this compound is reported to treat liver cancer and lung cancer, and some kind of significant results are shown here. Now, let me take you to the next slide about the adjuvant database. Okay, we also developed an adjuvant database. I think, hello, am I audible, madam? Yeah. So now let me take you to the next slide. That is about the adjuvant immunoanalytical database. Okay. So here, under my mentorship, the PG scholars of 2016 and 17 batch, it seems, they both have developed a database from C Sharp, SQL, and PHP. They developed a database for plant-based secondary metabolite adjuvants. Okay, this actually developed from Discovery Studio, of course. We have talked almost 1,000 compounds with most important target receptors of dendritic cells, and whatever 1,000 interactions we had, okay, we just posted in the database. Like if you want to get a plant adjuvant for a vaccine to be developed for a disease, for autoimmune disease, can be if you want to get a vaccine for arthritis, if you want to get a vaccine for the cancer, specific cancer, or for infectious disease, for all these things, okay, for any type of diseases, if you want to select a adjuvant, okay, you can use this database from the already docked interactions deposited in the database. Okay, of course, we have used these programs to deposit these kind of files. So I think I have completed to this. Then can we go? I think I can go for the second PPT. Yes, good. This is the second PPT. Or just we have just 4 slides. So let me complete these 4 slides. This is about the sponsored innovation project recently secured last month. Okay, last month our team has the Yukti team has received 5 lakhs. What is Yukti? Yukti is an innovation challenge, okay, organized by MHRD Innovation Cell, okay, especially we have Ministry of Education Innovation Cell, Government of India, and AST Government of India. The AST has sponsored us maximum of 5 lakhs for us to work on the development of a drug for hepatocellular carcinoma. Especially, I'm going to use a plant-based drug called Withaferin-A. Okay, I'm going to use the Withaferin-A, and I'm going to target a target receptor called mitochondrial HSP90. But here, I'm using nanotechnology. Other than the mitochondrial targeting technology, drug delivery, I'm also using the nanotechnology. I'm incorporating in that in addition, so that I can precisely target the disease tissues, inflammatory tissues when compared to the healthy tissues. So this is my objective. So of course, 135 innovations alone has secured grants out of 1,05,000 submissions. And this is a phase 3 pitching event. I just interacted with Dr. Dipan Sahu, who is an Assistant Innovation Director, MOE's Innovation Cell. Can I go to the next slide? Yes, good. Thank you. This is a product and process development of Withaferin-A happened in our lab, Department of Biotechnology, SVC. We have utilized the biochemistry lab, immunology lab, and genetic engineering lab. We have utilized so many labs to work on this. Of course, we also have approved animal house facility, CCSEA approved animal house facility. We are also working on animals at present. Okay, we have, of course, we got the seed from New Delhi botanical garden, and then we just grown this kind of plants in our soil, in SVC soil. We extracted the roots and we just powdered it, and we have gone for various extraction techniques. And after going for the rotary vacuum evaporator, we go on for the extraction and purification. Purification is we've done column chromatography. We have worked on HPLC. We're also going to purchase an FPLC presently to work to produce a drug commercially. And such a kind of a purified drug of up to 90 to 93%. We are using it to, we have checked with the in vitro studies in hepatic cell lines like a Hep G2 cell line and a Hep 3B cell line, and now presently working on mice. Okay. So, but we just tried with Withaferin-A, but also we are trying to use the nanotechnology, trying to also get a Withaferin-A tagged nanoparticles, and also try to work on these kind of researches in future. And now some sponsored projects and research consultancies happened in the past 2 years. Under IFTI, we got 5 lakhs to work on nanotechnology and mitochondrial targeting for hepatocellular carcinoma. So almost all these grants are we are working on the plant secondary metabolites. Okay, and if you see here, this second the DSD funded project, which I am as a principal investigator, I secured 18.3 lakhs. So of course, also working on Withaferin-A and Resveratrol, but on mitochondrial targeting technology. And from Sri Krishna, I just guided a few students. Okay, we got few grants in the form of a consultancy that I want to mean that we can all even the other engineering colleges, institutions, universities can also work in SVC under the form of consultancies. So SKAC and Welltech got profited on working on the projects. Also, we are getting some kind of good results from what we just working on the plant secondary metabolites, and even from the government of Tamil Nadu, we received grant, and even research fellowship is also getting a kind of a grants, okay, to work for the research fellow. They can also work on research fellowship. Yeah, next. So we are providing lot of opportunities to work on the funded projects. Okay, so anyone can also link with us to work on the projects, or even you can learn from me from the certificate courses, whatever we are showing it. And the next slide. So I think I have completed my presentation. Now it's up to you. So you can ask me.
Yeah, thank you so much, sir. This was a very knowledgeable session on the plant-derived secondary metabolites for treating various diseases. So the session is now open for questions. So the first question is, why is EdMed important for drug designing?
Actually, if you work on pharmacokinetics, in pharmacokinetics, majorly we have to rely on how much drug is utilized for treatment of disease. So if you take ADMET, A stands for absorption, D for distribution, M for metabolism, E for excretion, T for toxicity. In addition to the ADMET, we also can have L, like LADMAT, L element for liberation. So if a drug is taken in the form of a oral route, in the form of oral route, and from the buccal cavity, it goes and it reaches the digestive tract. So during the absorption, it happens in the blood, and the distribution is, of course, reaches to the desired tissue where we want to target. And metabolism is majorly happening in the cytochrome P450 enzymes of the liver. So if you see in metabolism in liver, nearly 50% of the drug is going to be utilized because of first pass effect. So only 50% of the drug, okay, only is going to reach the desired tissue. So you have to prepare a dose. Okay, so while preparing a dose, a number of doses, of course, you should have a knowledge of not only the ADME, but ADM, but also you should know the knowledge on E, excretion in excretion through kidney, excretion through urine, or in the form of a fetus on the sweat. So one should have a knowledge on the distribution of drug to the desired tissue by knowing about the ADMET properties.
Okay, sir. The next question is, which is the best method of drug delivery of quercetin for high bioavailability?
So everyone defaultly have prescribed or preferring to use the oral drug delivery. Okay, because even can take kids to the kids to the elders. Okay, most of them they don't want to take intravenous injections. We better prefer the oral route. Okay, if so, but the oral route won't give you the high bioavailability. But still, these kind of a plant compounds, if you want to reach the high bioavailability, you have to use drug delivery technologies like nanotechnology. You can incorporate the nanotechnology, whatever I mentioned earlier. Use nanosponges or nanoparticles. So there are different nanotechnologies available. You can choose any nanotechnology which is suitable for a compound like quercetin. And also you can integrate with other technologies like mitochondrial targeting. I have mentioned, for the cancer tissues, I express more uh, in the mitochondria. The cancer tissues have more mitochondria, and so the expression is more. So it can target the organelles like mitochondria for the efficient drug delivery, which gives a high bioavailability.
Can the same method be adopted for designing any drugs?
Yes, sure, but you have to create the synthetic libraries. I'm not using a drug as such. I'm also integrating with mitochondrial targeting region. So one has to check even with how whether is it working with the medicinal chemistry persons or not. Okay, you have to check whether this can be synthesized or not. If it is possible, you can try, or you just check with your plant compounds whatever you expected.
Okay. The next question is, what are the benefits of natural products in treating diseases?
So in earlier times, okay, in modern times, if you see many people go for only these kind of Ayush approaches, Ayurveda, Siddha, Unani, etc. Okay, so our grandfathers might have, even my grandfather is who sell the medicines from the herbs. Okay, just give in the form of a herbal medicine in the form of chornum, etc. He cultivate plants in his own land and he try to produce. So no one at that time can go to the doctor if the expenses are more. So generally they take only the plant. They take the drugs only derived from the plant sources. So we have numerous benefits from the natural plants. Uh, uh, we can not only see from the Ayush, but also you can come to know even on the website itself. I mentioned so many side effects are reduced if you try to take natural products. Okay, in the synthetic drugs, you have to encounter, you have to combat few more kind of diseases. So it's a very difficult. The natural products can target many pathways, and obviously I can tell you just because it is targeting most of the disease pathways, this is possible to reduce the side effects when compared to synthetic allopathic drugs.
The next question is, what is the most important marker for authentication of medicinal plants?
So of course, I told you my database, the database, you remember, initially I told IMFAT I mentioned. So that is the best website. Of course, also mentioned by the first speaker. Okay, even I can suggest you to go for the IMFAT before going for any drug design research, including vaccine adjuvant drug research, drug vaccine adjuvant research.
How to bridge natural products with modern drug designing?
So natural products, of course, you are developing. I can suggest you to isolate a specific compound. You know, if you search in India, I am working on Withaferin-A compound and also Withanol compound. But when I when I try to search in India Mart, okay, which is the major uh, industry for, which is the major vendor for providing us the product, they are providing a kind of an extracts in the form of ethanoids. They are marketing like ethanoids, but that is nothing in extract. In extract, you can find not only the ethanoids but also other compounds that won't give you the exact results. Okay, some compounds, some secondary metabolites can inhibit the same metabolites. That's what I think you can got the information from the first speaker, from Ayush. She has mentioned whatever metabolites derived during the synthesis can inhibit the other potential compounds. If it inhibits, then obviously you can't tell what is the effect of a specific compound. So in my research, whatever I'm working is on a specific compound. I either work on Withanol A or I work on Withaferin-A or I work with Praveen. Then only you can get the exact results. If you work on series of compounds, it will be very difficult. So I better suggest you to go for structure-based drug design instead of traditional drug design. Instead of using this chorna kind or a kind of an Ayurveda formulations, better go for purifying a particular compound and try to work on it. So go for go for structure-based drug design. Then you can go for the synergistic effect. You know, the Withanol A and Withaferin A or the plant compounds derived from ashwagandha. These compounds are used as an adjuvant for preparing a vaccine. And this kind of research was completed by Serum Institute of India in 2010. They got a meningococcal polysaccharide vaccine, okay, in 2010. I believe Professor Suresh has brought this kind of, Dr. Suresh has got this kind of a product, a plant-based adjuvant for meningococcal polysaccharide vaccine. He has used separately Withaferin-A and Resveratrol separately, and also used in the combination. He got good results even in combination. Withaferin-A and Resveratrol can also used as a best plant-based adjuvant for treating meningitis. Okay, so that's what can able to find. So we have to one have to bridge this even compounds, and then you can check how effectively it works even in structure-based application.
There's another question. Can you suggest a best source material for literature on plant secondary metabolites you have come across, sir?
Yeah, I think this session is already recorded in YouTube video. So you just, I think it will be shared, isn't it, madam? So if it is
Yes, sir.
shared, obviously you can find some 11 databases I've shown earlier. I think in the fourth or fifth slide I've shown about a list of plant secondary metabolite databases. So the participants can see that. Okay, so relaxedly you can pause and you can check and you can work on it. I think I've already provided this information in the PPT itself.
Yes. Sir, so it looks like we are done with our questions.
Good, thank you.
So thank you so much, sir.
And
So
also,
it's time to finish. Yes,
yeah,
sir.
I also love, finally like to thank you for hosting. Also, I like to thank Ms. Divya Shanmugarajan, madam, who has given me an opportunity to interact with the outside world. And also, I thank the higher officials, the management, and even the faculties and non-teaching staffs who supported me in my research. Okay, so I thank even Almighty and my parents for presenting a valuable topic of utilizing the plant-derived secondary knowledge for the treatment of disease. Thank you.
Thank you, sir. So it's now time to facilitate our speaker of the day with a memento and certificate of appreciation.
Thank you,
So
madam.
thank you so much, sir, once again for giving your time. And it was really a pleasure to have you with us here today.
Can I leave?